Today's lesson we're looking at photosynthesis, and the photosynthesis that we're looking at is specifically for C3 plants. Don't worry about the C3, C4, C10 plants yet. Essentially, they have a very similar photosynthesis system, but there are minor changes that we will be discussing in the next lesson. For this lesson, we're looking specifically at C3 photosynthesis, the entire thing. And then when we get to the other plants, I'll tell you where it differs in terms of its function. Any questions before we start? Yes, recording. Sure. Yeah, recording. All right, let's move on. So, first question, right? What is the name of this structure? This is a diagram of a chloroplast. And remember, I told you inside a chloroplast, there are like pancake structures. This is where one part of the photosynthesis reaction occurs. The one that we talked about, the one that requires light. What is the name of that structure? Can I get Zoe? What do you reckon? It's a T. Yeah, definitely something with T. I'm pretty sure we put pictures to those owners in the review question. Okay. Yeah, I don't. Does anyone actually know? Abhi? Yeah, I feel like it's been a while since we even did that short review lesson. So these are referred to as thylakoids. Yeah, the thylakoids, this is where the light-dependent reactions occur. So that means where light is required for the reaction to occur, that will happen at the membrane of the thylakoids. The second stage of the reaction happens in the space between the thylakoids and the chloroplast. So the stroma, we call the space stroma. So that's where the second reaction happens. But the first reaction, it happens at the membrane of the thylakoid. Alright, today's lesson, you guys are going to learn about photosynthesis as an overall process, the two stages that are involved in it, light-dependent and light-independent reactions. You will learn about the inputs, outputs, and where it happens. So in each stage, a different reactant or a different product might be released. For example, H2O is required in the first stage. CO2 is required in the second stage. Sunlight is required in the first stage. Glucose is only released in the second stage. H2O is released in the second stage as well, but oxygen will be released in the first stage. So you can see that if you break down the overall equation of photosynthesis, you can see that different reactants are used at different points and different products are released at different points as well. So this is a generalized equation for the entire process, but when we actually learn in detail, we know that there are two stages to it. So we need to know that which products are released at which time and which reactants are used at which time. Another thing you should know is the biochemical pathways of photosynthesis. Remember, when we see biochemical pathways, we're referring to the pathways that are created by different enzymes that are breaking down molecules or building up molecules to get a final product. So an example would be something like your digestion in your body. We start off with carbohydrates, and then different enzymes break it down into smaller and smaller components until we get to glucose. In plants, there is a similar biochemical pathway, but for the purpose of photosynthesis. So there are actually a bunch of enzymes that work together to make sure they can carry out photosynthesis. And input, output, generalize. So you'll need to know the general role of the enzymes and also the coenzymes involved. Remember, the coenzymes are ATP and the NADPH. Those are the coenzymes. You need to know where they come in. You need to know the input, output, and the general structure of biochemical pathways. Right? Moving on. So here's the glossary task. And do you guys want to watch the video we watched on Monday again? Or we can move on to the content. Move on. All right. So we start off with the sun, right? The sun is the biggest energy source in the solar system, as we are very aware of. So early life was trying to use this sunlight, use this energy coming from the sun to make food for itself. And that's when evolution gave rise to the first cell that is able to do photosynthesis, which is a cyanobacteria. And over time, evolution allowed plants to emerge that can also do photosynthesis. But what these living organisms are doing is essentially converting the solar energy, the light energy coming from the sun, into usable energy for themselves. So for plants, it would be chemical energy in the form of uh, glucose that is being produced here. One thing that we do is convert the solar energy into electrical energy using solar panel. So if you really can't remember photosynthesis, just think of it as plants' way to do solar panel charging, right? So photosynthesis can be broken down into two parts. Photo meaning that it's light, synthesis meaning that it is creating something. So essentially, this process is referring to creating something out of light, essentially putting together glucose. What it does is that plants, specifically the leaf section of plants, trap that sunlight. It's in the form of chemical energy and stores it in bonds of glucose. That is the most simplified definition of photosynthesis, where plants absorb the sunlight and convert it into chemical energy in to be stored in the bonds of glucose. So when we refer to an organism that is able to do this, not only plants but also the cyanobacteria that I referred to, they are going to be called as photoautotrophs, meaning that they can make their own food using photo energy, light energy. That's why it's called photoautotrophs. So the word autotroph refers to something that can make its own food. So when it says photoautotroph, it's referring to the fact that it makes its own food using sunlight. So, like I said, yeah, overly simplified, and there are different stages involved for the reactants and the products. So we're going to look at the first stage. Okay, first let's talk about where it happens. So when we think about photosynthesis, it generally happens in one organelle within the cell, which is the chloroplast. So you guys would have learned about plant cells, the organelles within a plant cell, so nucleus, mitochondria, Golgi apparatus, endoplasmic reticulum. Those are all the common ones. The one that is unique to plants and living organisms that can photosynthesize is chloroplast. That is the organelle that is essential for photosynthesis to occur. So essentially, if any cell has chloroplast, you can conclude that it is definitely undertaking photosynthesis. Inside that photosynthesis, there is a pigment referred to as chlorophyll. So it's a green pigment that basically absorbs the sunlight and stores it to be used later on in the chemical reaction. So I know we've talked about photosynthesis as being plants absorb sunlight, they get glucose, but the process is much more complicated than that. Essentially, how it starts is that sunlight needs to be stored within the plants to be used later for a chemical reaction, and chlorophyll is in charge of storing that energy temporarily. So sunlight is absorbed at the leaves, chlorophyll absorbs it, keeps it in the form of chemical energy, and then uses it later in the reaction to then make glucose. So this reaction, so like I said, plant cells, so any cell with chloroplast is able to do photosynthesis, but not all cells will do photosynthesis equally, right? For example, like I said, right, plant cells, all of them would most likely have chloroplast, but not all plant cells do photosynthesis at the same rate. For example, if I look at the cells at the stem, they are also doing photosynthesis, but compared to the leaves, they are doing much lesser. So the leaves are where most of the photosynthesis is going to be happening. So the bulk of the photosynthesis carried out the plant is carried out at the leaves because the leaves have cells known as mesophyll cells that have a high concentration of the organelle referred to as chloroplast. So chloroplast conducts photosynthesis, and wherever there's a high concentration of it means that's where most of the photosynthesis is occurring. And where can we find the high concentration of mesophyll cells? Is at the surface of the leaves, right? So if this is the surface of the leaves, the mesophyll cells will be directly underneath it so that it can get maximum sunlight hitting it and absorb it to create glucose. Another thing that is important for the plant to conduct photosynthesis is carbon dioxide. That is one of the reactants that you can see in the chemical formula. How does the plant obtain this? Is through the stomata. So you can see this, the stomata are basically tiny pores within the bottom part of the leaf that allow for the gaseous exchange to occur. So in this example, so in this context, it is absorbing in carbon dioxide. So it's taking in carbon dioxide from the atmosphere for the process of photosynthesis. But the stomata is actually used to release oxygen as well, and also to release H2O that is produced as a product of photosynthesis. The second thing that's required for photosynthesis to occur is water, which you can see in the chemical formula, and that is obtained through the root hairs, right? So basically, plants get water from the roots. That water travels up the stem in the xylem to get to the leaves, where it will supply the cells with the water required for photosynthesis. So these are the three requirements for photosynthesis, right? Water and carbon dioxide are the reactants, whereas sunlight is just a prerequisite. It is very important that you do not state sunlight as a prerequisite molecule or prerequisite reactant, because it is not the same as water and carbon dioxide. It is just a prerequisite, but it's not a molecule or anything like that. So do not refer to it as such. So these are the two requirements and how they obtain it. So if we look at the stomata, if you zoom in, this is what you're going to see, right? These are tiny pores that can open and close depending on what time it is and whether the plant needs to actually open and close it. So when it is open, CO2 can go in, oxygen can come out, and so can H2O. And this is the normal process. But when it's closed, CO2 come out and CO2 can't go in and oxygen can't come out either. This is an important thing to note. When the stomata is closed, no gaseous exchange will occur within the plant. So whatever's inside will stay inside, and whatever's outside will stay outside. The next thing is the root hairs. Let's try to zoom in. So these root hairs, they absorb water, and then it travels up the roots in a structure known as the xylem. That is the structure used to transport water within a plant. So the plant absorbs the water and it's traveling up the xylem until it gets to it gets to the site of photosynthesis, where it will undergo the reaction. Are we good here so far? So we have the reactants, and we know that the mesophyll cells, which are the cells that are directly underneath the leaves, that's where most of the photosynthesis occurs, because that's where the highest concentration of chloroplast is. Chloroplast is the organelle that does photosynthesis. So without it, no photosynthesis occurs. So now that we know where and what's happening, let's see the actual stages involved in photosynthesis. There are two stages that are important to know. The first stage is the light-dependent reaction, and then the second stage is the light-independent reaction. Within the names itself, you already know one of the features, which is that the first stage definitely requires light to be carried out. That's why it's depending on the light. The second stage, however, is independent from the light, meaning that it doesn't need the light. So it can do the second stage whether it's in the light or in the dark, it doesn't matter. But that's the case about it. It doesn't need light to carry out this reaction. The reason is very simple. Basically, the energy absorbed in the first stage is used to carry out the second stage and create glucose. So let's look at the first stage. So like I said, it depends on the light, and it occurs at the thylakoid membrane of the chloroplast. So if this is one chloroplast organelle, and we zoom in, we will see the thylakoids that are stacked together like pancakes. And if you zoomed in, this is what you would see, the structure of that. So I know this looks very complicated, but we'll go one step at a time. Then the video will help explain. Okay. So essentially, what happens is when light makes contact with the thylakoid membrane, it triggers two reactions. One is the excitement of electrons. When the electrons here get excited, they will start moving along something known as an electron transport chain. So you can see the pathway that the electron will take, but the first stage is that it starts to move and gets to the first membrane protein. This is a hydrogen pump. Okay, hold on to that idea first. That's the first reaction. The second thing that happens is also H2O will be split to replace the electrons that are moving here. So when these electrons move, this membrane protein will be lacking in electrons, and that is a problem. It always wants to have the same number of electrons. So the H2O will be broken down through the action of light, and the electrons from that will go into this, and the hydrogen ions will flood inside the thylakoid. So it's important to note that the hydrogen ions are accumulating inside the thylakoid. And then you see H2O, there's hydrogen, and then there's oxygen as well. That oxygen is where the first product is coming from. So as soon as this is separated, it is released, and it is released from the plant through the stomata. Like I said, gaseous exchange occurs. So that's the first stage where excitement occurs, electrons move, H2O is broken down, hydrogen ions are inside, oxygen is released. So now what happens is, as the electrons move across this hydrogen pump, it powers the hydrogen pump. And you can guess what a hydrogen pump will do. It pumps more hydrogen inside of the thylakoid. So the whole point is to get a very high hydrogen concentration inside here. There's two ways it's done: through the hydrogen pump and also through the breaking down of H2O. So when there is a high hydrogen concentration in here, that will make it want to go to a place where there's low concentration. Essentially, in chemistry, if you haven't learned this, high concentration gradients don't like to be in a high concentration gradient. They like to equalize. So they will always head to the low concentration area. So because there's such a high concentration area here, high concentration gradient of hydrogen ions, they will want to go to a place where it's low concentration, which is outside, and there's only one way for it to happen, which is through the ATP synthase. So it allows for hydrogen to flow out, but as a bonus, what happens when the hydrogen goes out is that ATP synthase will start to spin, creating ATP. So it uses the energy of the hydrogen ions flowing to convert ADP into the charged version as ATP. That is the first charged coenzyme that is going to be produced in the light-dependent stage. Remember that there are two coenzymes that are made. One is ATP, the other one is NADPH. So the first one's already made through the action of ATP synthase. Now let's talk about how the second one is made. So after the electrons from here have gone through the hydrogen pump, they will continue moving along until they get to the third membrane protein. This is the site where it will be, the electrons will be used to convert NADP plus and hydrogen ions into NADPH, which is the second charged coenzyme. These two coenzymes are the reason why the second stage of photosynthesis does not require sunlight. They essentially carry the energy for the reaction to occur, so no energy is required from the outside because all the energy is already ready to go. Anyone? Okay, I'll just show a video on this because the video animation always makes it much easier to understand. Essentially, if I had to simplify that entire thing into this one picture, you can see that, right? Sunlight comes in with water, and then the uncharged coenzymes are becoming charged through the reactions to become ATP and NADPH, and oxygen will be released as a product. Right. Let's have a thing. Yes.
In photosynthesis, light is used to make organic compounds, especially sugars, in plants.
This is extremely important for virtually all life on Earth, because these organic compounds are what we and our food are all made of. So how does this process work? In short, when light strikes a leaf, this light is absorbed and the light energy is used to drive a series of chemical reactions that ultimately make sugars and other organic molecules. Overall, carbon dioxide and water are taken in by the plant and used to make glucose and oxygen gas. Photosynthesis can be divided into two sets of reactions. One, the light-dependent reactions, and two, the Calvin cycle. The light-dependent reactions, as you might have guessed, depend on light. To understand how these work, let's zoom in on a leaf so we can see them happen. Leaves are made of plant cells, and inside of these cells are special organelles called chloroplasts that do photosynthesis. Each chloroplast has many discs called thylakoids with pigments, like chlorophyll, that absorb light. Think of thylakoids as solar panels in the chloroplast. They absorb the light energy from the sun. Now let's zoom in on the thylakoid and focus on the thylakoid membrane. This is where pigments such as chlorophyll are found and is where light gets absorbed. The pigments are arranged in clusters called photosystems. Let's start with photosystem 2. When photosystem 2 absorbs light, electrons in chlorophyll gain energy or get excited from the light. These excited electrons leave chlorophyll and move to something called an electron transport chain. But since chlorophyll lost those electrons, they need to be replaced. To replace them, water is split and its electrons go to chlorophyll. When water is split, electrons, hydrogen ions, and oxygen are produced. You probably know that plants make oxygen when they do photosynthesis. This is where that oxygen comes from. Now, back to the electron transport chain. The electron transport chain transports electrons. As it does, the energy in the electrons is used to pump hydrogen ions, or protons, across the thylakoid membrane into the thylakoid. This creates a high concentration of hydrogen ions inside the thylakoid. These ions would be trapped inside the thylakoid, but a special enzyme called ATP synthase allows them to passively diffuse from high to low concentration. This flow of hydrogen ions through ATP synthase causes ATP synthase to spin and produce ATP, similar to how water flowing through a turbine produces power at a hydroelectric dam. This ATP is a key product of the light-dependent reactions. When the electrons reach the end of this first electron transport chain, they go to photosystem 1, where light excites them once again. They travel down a second, shorter electron transport chain, where they are accepted by a molecule called NADP+. When it accepts the electrons, it also accepts hydrogen and becomes NADPH. NADPH is an electron carrier and is another key product of the light-dependent reactions. It carries electrons and hydrogens to the next set of reactions in photosynthesis, the Calvins. Both ATP and NADPH are critical products of the light-dependent reactions that are needed to make sugar in the Calvin cycle, which we will examine in our next video. If you'd like to try the light-dependent reactions yourself, check out the link for the Photosynthesis Interactive at Bioman Biology. And don't forget to subscribe if you'd like to be notified when I release new videos. This flow of hydrogen ions through ATP synthase causes ATP synthase dependent reaction. They travel down a second, shorter electron transport chain.
Alright. Hopefully that video kind of made sense to you guys. We'll go through it one more time. Alright, so it all starts with sunlight entering the system. It will be exposed to the photosystem 1, and that excites the electrons in the system, making the electrons move along the electron transport chain. When the electrons start moving, the missing electrons need to be replaced, and that's where H2O comes in. And H2O will undergo a process known as photolysis. Okay, that's where it's breaking down the H2O using photo energy to increase the hydrogen concentration within the membrane and also to supply electrons to the photosystem. Then the oxygen atoms are released into the atmosphere to be given out as a product of photosynthesis. So when the electrons move across the electron transport chain, they will eventually power a pump to increase the hydrogen ion concentration within the membrane. while also eventually getting to another membrane protein where it will bind together with NADP plus to become NADPH. Okay, that is the charged version. Okay, NADP plus is the uncharged, NADPH is the charged version. That is one part completed. Now the second part is when there is a very high hydrogen concentration within the thylakoid lumen. That high concentration wants to become equal with the exterior. So what happens is hydrogen tries to flow out in whatever way it can. And the only way it can at this point is through the ATP synthase. And every time it moves along the ATP synthase, it will make the ATP synthase turn, and that energy is used. So that energy will be used to create ATP from ADP. So ADP is the uncharged version, ATP is the charged version. So two coenzymes are made during the light-dependent reaction, the NADPH and also the ATP, and then oxygen is also released as a product of this reaction. Everyone keeping up with me so far, right? All right, so moving on.
In photosynthesis,
Yeah, you don't need to read through that. So essentially, yeah, that's what we looked at, right? We need 12 H2O for this process to occur. That's where this the H2O that is being broken down here through photolysis, that's why it's needed. The 12 NADP plus, that's the uncharged coenzymes, they are also needed for the reaction. And another thing that's required is the 18 ADP molecules that are going to be converted into ATP. So since we need to have the charged coenzymes at the end of the reaction, we need the uncharged versions as a reactant for it to be converted into the charged version. So once these uncharged coenzymes are charged to become NADPH and ATP, we can then release them into the stroma where it will facilitate the second stage of the reaction, which is the light independent reaction. Keeping up so far? All good? Right. If at any point you guys are not following or it's becoming a bit heavy, just let me know and then I will do a short recap. All right, so let's talk about the role of enzymes and coenzymes in the photosynthesis. So I constantly mentioned the fact that this is a coenzyme, that NADPH and ATP are coenzymes. But what is a coenzyme, right? Coenzymes are factors that can influence the action of an enzyme, and they are they are required for the activation of an enzyme. So essentially, if you see this picture down here, you see the picture down here, you can see that for an enzyme to be able to bind to its substrate, a coenzyme or a cofactor needs to first bind to the enzyme. Only once it has bound to the enzyme, it will activate the enzyme and then making it complementary for the substrate to bind. If the coenzyme never bound to it in the first place, the substrate won't be complementary to the enzyme, meaning that the substrate won't undergo catalysis and it won't be broken down. So remember, cofactors or coenzymes are essential for the activation of an enzyme. So in this case, the coenzymes NADPH and ATP are essential for the next stage of photosynthesis to occur. So if these molecules are not made, then photosynthesis will be dead in its tracks and it won't occur moving forward. So these coenzymes are pretty much reusable. They're not broken down once they're used. This two coenzymes can be used over and over and over through the light dependent and the independent stages. So essentially, at the light dependent stage, there's energy present. So when the uncharged particles are supplied to the light dependent reaction, they are returned back as charged particles. So NADP plus comes in to the light dependent reaction, goes back as NADPH. ADP comes in the light dependent reaction and goes out as ATP. They are both going into the light independent reaction to be used, and when they're used, they're returned as uncharged particles. This cycle will continue over and over. They become charged and then they become uncharged. Charged, uncharged. And then they will cycle through one after another over and over, because like I said, coenzymes and enzymes can be used over and over and over. They do not get broken down when they are used. All right, moving on. So now let's go into the second stage of photosynthesis, which is the light independent reaction. Like I said, it's called light independent because it is independent from the light. It does not require light to carry out this process. So essentially, it can happen whether there's light or no light. That's not a factor in its function. The reason why is that the energized coenzymes ATP and NADPH provide the necessary energy for this reaction to occur, so light is essentially not required. Another thing to note is where the reaction happens. So let me zoom into this picture because it shows a much nicer stroma. So you can see here, this is the structure of chloroplasts again. And then we have our pancakes. We have our pancakes, which are the thylakoids. And then we have the space between the thylakoids and the membrane, which is called the stroma. That is where the second reaction occurs. So the first reaction happens at the membrane of thylakoids, whereas the second reaction happens at the space outside, which is the stroma. But this is the important thing to note. Both reactions happen within the chloroplast. It's just within the chloroplast, there are different sites for each reaction, but both happen inside the chloroplast. The first one will happen at the membrane of the thylakoids within the chloroplast, and then the second one will happen at the stroma within the chloroplast. Do not ever confuse that. Chloroplast is the site of photosynthesis, and that does not change ever. Okay. So let's talk about the reactants required for this reaction. So as I mentioned here, the first H2O is used up in the first stage. So the second reactant, which is CO2, is needed for the second stage reaction for a process known as carbon fixation. Associate the idea that carbon dioxide is related to carbon fixation because this is going to be the same for C3, C4, and also CAM plants. So essentially, by remembering that CO2 is associated with carbon fixation, it will save you some time. The second thing, the two other things that are required are the coenzymes, which we need 12 NADPH and 18 ATP. So with the numbers, it is somewhat important that you know the distribution of the numbers. One thing I can recommend that might make it easier is the number of NADPH and the number of ATP will be the exact same as ADP and NADP plus, because it keeps going in a cycle, so the same amount is used over and over, right? If 12 NADP plus is used, 12 NADPH is used, 12 NADP plus is given back. If 18 ATP is used, 18 ADPs are given back, and so on. Another thing that's produced in this stage is obviously glucose and water, the final two products. So that pretty much summarizes your entire photosynthesis reaction between which stage, which reactants and which products are produced in which stages. So now let's look a bit further let's look at it with a bit further more detail.
The Calvin cycle is the second set of reactions in photosynthesis and occurs after the light-dependent reactions. Sometimes the Calvin cycle is called the light-independent reactions, or even the dark reactions, because light is not directly needed to make the Calvin cycle happen. Nevertheless, two key products from the light reactions are needed to make the Calvin cycle work: ATP and NADPH. You may recall that both ATP and NADPH were made during the light reactions at the thylakoids. The Calvin cycle takes place in the fluid surrounding the thylakoids. atoms, known as the stroma. ATP provides the energy, and NADPH provides the electrons and hydrogens that are needed to reduce carbon dioxide, build sugars in the Calvin cycle. Here's how it works. Phase 1, carbon fixation. An enzyme called Rubisco takes CO2 from the air and adds it to a 5-carbon compound called RUBP. This is called carbon fixation. This makes a 6-carbon compound that immediately splits in half to make 2 molecules of 3-phosphoglycerine. Notice that we did carbon fixation for 3 molecules of CO2. You will see why soon. Phase 2, reduction. In the reduction phase, ATP from the light reactions provides energy to make an intermediate compound. This reactive intermediate can then be reduced or gain electrons and hydrogen. The electrons and hydrogen come from NADPH and are used to reduce the intermediate compound to make glyceraldehyde 3-phosphate, more commonly called G3P, or sometimes triose phosphate. G3P is the key product of the Calvin cycle that can be used to make sugar. Notice that for every 3 molecules of CO2 brought into the Calvin cycle, there's a net gain of 1 G3P. 3 carbons in, 3 carbons out. Makes sense, right? Phase 3, regeneration. Notice in our model that only 1 G3P is removed from the cycle, but 5 more remain. These 5 G3P molecules get converted back to our starting compound, RUBP. Remember, the Calvin cycle is a cycle, so we need to make our starting compound again to keep the cycle going. Now, I'm going to give you a moment to think. If we have 5 G3P molecules, and each one has 3 carbons, how many carbon atoms do we have? Now that you have the answer, how many 5-carbon RUBP molecules can we make using those carbon atoms? I'm guessing that you figured it out. We can make 3 RUBP molecules. To do this, energy is required. Can you guess where that energy comes from? You said ATP. You are right. Let's watch. Now that we've completed the Calvin cycle for 3 carbon atoms, let's go through the process again so that we can make glucose. Remember, our overall photosynthesis equation has 6 carbon dioxide being used to make 1 glucose molecule with 6 carbons. With this in mind, how many G3Ps are needed to make 1 glucose? You said 2. You are correct. Now we've seen how the Calvin cycle is used to make glucose, and how light-dependent reactions provide the NADPH and ATP that were needed to make the Calvin cycle work. The glucose and other organic compounds made as a result of these processes in photosynthesis are what allow plants and algae to use carbon dioxide being used to make 1 glucose molecule with 6 carbons.
All right, I can imagine how heavy that seems. So let's go through it one step at a time. So essentially, if you broke this down if you broke down the Calvin cycle into its separate phases, there are 3 separate phases that are distinct from each other. The first one is called carbon fixation. Okay? This is basically where carbon dioxide is fixed in a way for it to become a 3-carbon molecule. So this is why this is referred to as a C3 plant. This exact stage right here, where it generates a 3-carbon molecule. That's how it's different between the other 2, C4 and CAM. C4 and CAM make a 4-carbon molecule here instead of a 3-carbon molecule. So that's how they're different, but we'll get to that later on. But here, the first stage, carbon fixation, a molecule known as RUBP will bind together with Rubisco along with carbon dioxide that's coming from the outside. RUBP, one thing to note, has 5 carbons attached to itself, and carbon dioxide has 1 coming from outside. So when when both of them bind together, we'll have a total of 6 carbons that are binding to this one enzyme known as Rubisco. This enzyme is actually incredibly important for the function of photosynthesis, and in essence, this is actually the most common enzyme in the world. So I'm not sure in terms of concentration how much it is, but yeah, it is definitely the most common enzyme in the world, because without this, photosynthesis can't occur. So you can imagine every plant in the world needs this enzyme to be able to carry out photosynthesis. That is why it is a much more abundant enzyme than anything humans make. Okay, so this enzyme allows RUBP to bind to it, allows carbon dioxide to bind to it, and once they bound together, catalysis of reaction occurs where a 3-carbon molecule is generated. This 3-carbon molecule is referred to as these names.
H. Pushing the carbon fixation for 3 molecules of CO2. You'll see why soon. Phase 2, reduction.
Alright, essentially with these names, right? 3-phosphoglycerate, you will not need to know these full names. Lucky for you. You'll need to know their short forms though. Okay, I will talk about the short forms in a bit. The next picture, they're all available. But for now, just remember that Rubisco catalyzes a reaction where 6 carbons are then reacted in a way to become 2 3-carbon chains. So each chain will have 3 carbons attached to themselves. So since there were 6 carbons that are reacting together, a total of 2 chains will be made. Each chain having 3 carbons. So are we following so far, right? 5 carbon comes from RUBP, 1 carbon comes from carbon dioxide, they bind to Rubisco, and then Rubisco will fix the carbon in such a way that it becomes a 3-carbon chain. And since there were 6 carbons here, it will make a total of 2 chains of this phosphoglycerate molecule. Right? Once this process occurs, the next stage is called reduction. This is where the charged coenzymes come into play. So the first thing that happens is that ATP will react with the phosphoglycerate to basically provide energy, and then another thing will occur where NADPH will react with it
In the reduction phase,
reduce
ATP from the light reaction provides energy to make.
So there's 2 things that occur, right? One is where energy is supplied, and the other one is where the electrons are used to reduce the molecule into another molecule. So if you haven't learned this in chemistry, electrons and reduction and all that, don't worry about that. Just refer to it as reduction has occurred using NADPH. That's all you need to say. Right. Reduction has occurred using the action of ATP and NADPH. But if they ask specifically which coenzyme, you would refer to as NADPH, because that is the enzyme that carries the electrons to allow the reduction to occur. ATP only carries the energy to facilitate the transition for it to become an intermediate molecule. So you don't need to know about this intermediate molecule. Essentially, for this stage, you would say that both ATP and NADPH is used to reduce the phosphoglycerate molecule into the G3P molecule. But if they ask specifically, you would say NADPH is the one responsible for it.
An intermediate compound. This reactive intermediate can then be reduced or gain electrons and hydrogen. The electrons and hydrogen come from NADPH and are used to reduce the intermediate compound to make glyceraldehyde 3-phosphate, more commonly called G3P, or sometimes triose phosphate. G3P is the key product of the Calvin cycle that can be used to make sugar.
All right. So this is the first. Yeah. So once that reduction has occurred, 6 of these G3P molecules are first accumulated before moving on to the next stage. Okay. For each cycle, there are a certain number of molecules that are produced. You don't need to know that. All you need to know is that how many are going where. So 6 is produced here, G3P. And the thing to note is that not all 6 will be converted into glucose. Not all 6. In fact, only 1 is kept aside to be made into glucose, whereas the other 4 are actually sent back into the cycle to be made back into that molecule that we talked about in the start, which is RUBP. Okay, that RUBP will continually be produced and used up in this Calvin cycle in the same way that we talked about the coenzymes being used in the overall cycle as well. So RUBP is also going to be used in a cycle where it's used for carbon fixation, and then once it's been reduced to G3P, 5 of those are taken back and made into RUBP again. So we can look at that.
Notice that for every 3 molecules of CO2 brought into the Calvin cycle, there's a net gain of 1 G3P.
So yeah, essentially, for every 3 carbons that come in, only 1 G3P can be made because 1 G3P has 3 carbons, right? So how many carbon dioxide atoms do you think will be required if you want to make 6 G3Ps? So think about it. How many atoms of carbon does a carbon dioxide have? 1, right? And then Rubisco supplies the other 5. So if you want to make a total of 6 G3Ps, and each of them has a total of 3 carbons, how many carbon dioxides would you need to make 6 G3Ps? 6. Yeah. Because the rest of all the carbons are going to be 5, 6. No, it's 3. Okay, I was confused. No, that doesn't make sense. Because think about it. Let me try to simplify this. So Rubisco has 5, right? 1, 2, 3, 4, 5. Carbon dioxide has 1. So in total, there would be 6 carbons when both of them are combined together. 1 G3P has 3 carbons. So essentially, for every time that Rubisco and carbon dioxide are fixed together, it makes 2 G3Ps eventually. So if you want to make 6 of this, we just need 3 of these things to happen. 3 carbon fixation to happen, and it will generate enough to make 6 G3Ps.
3 carbons in, 3 carbons. Makes sense, right? Phase 3, regeneration. Notice in our model that only 1 G3P is removed from the cycle, but 5 more remain. These 5 G3P molecules get converted back to our starting compound, RUBP. Remember, the Calvin cycle is a cycle, so we need to make our starting compound again to keep the cycle going. Now, I'm going to give you a moment to think. If we have 5 G3P molecules, and each one has 3 carbons, how many carbon atoms do we have? Now that you have the answer.
Alright, that one, that one's the next stage. So let me come here from reduction, right? So let's start the cycle again. I'm going to go through it fully. So when the cycle phase 3 is finishing, you will have RUBP made back again. RUBP is the molecule with 5 carbons attached to itself as a chain, and that will bind into Rubisco, taking up these 5 spots. Carbon dioxide will come as one of the reactants of photosynthesis and bind here as well into the last spot, making it a complete making it such that the enzyme can actually catalyze the reaction of carbon fixation. When that happens, it creates a molecule known as 3-glycerate. Glycerate, something like that. The short form I'll let you know later. That is the first molecule generated that has 3 carbons. Okay, that needs to be reduced to become into G3P. So how is it reduced? Through the action of the coenzymes ATP and NADPH. When that happens, it becomes G3P, and this is done 3 times to make sure a total of 6 G3Ps are made, and only then the next stage occurs where 5 will be sent back into the cycle to be made into RUBP. The regeneration of RUBP, 5 G3P molecules are going to be taken up for that, and 1 will be put aside to be made into glucose. So here's a little question for you guys. If this has 3 carbons, this G3P has 3 carbons, is one of this enough to make glucose? Can I get it? Okay, if this is 1 G3P molecule, okay, it has 3 carbons, right? Is 1 enough to make glucose? Why? So how many carbons does glucose have? 6. And this one has 3, right? So obviously you need 2 of these. So that's the key thing here. This cycle as well needs to accumulate 2 G3Ps for it to be converted into glucose as we know it. So here we need 6 G3Ps in total for the cycle to continue, where 5 will be sent through and 1 will be accumulated here. But even here, 2 needs to accumulate before it can be converted into glucose. So overall, this cycle repeats itself over and over a lot just to generate enough to make glucose. Any questions so far? All right. So once that's happened, there's been 2 G3Ps stored away to make glucose. 5 will be sent back, and it will be regenerated into RUBP. And this is done through the action of ATP again. Remember that the distribution of ATP to NADPH, only 12 NADPH was required, whereas 18 ATPs were required for this reaction. Why did it require so much ATP? It's because ATP is used here and also in this stage. So there's 2 places where ATP is required. That's why 18 ATPs are used. But whereas NADP is only required here, that's why 12 of that is enough for this reaction. So when ATP reacts together with G3P, it will make 3 RUBP molecules. Why does it make only 3 RUBP molecules from 5 G3P molecules? Can I get Nyaput? Okay. So G3P molecules, there's 5 of them being sent back to be made into RUBP, but only 3 come out. Why? It has something to do with number of carbons in the molecules. How many molecules of carbon does RUBP has? So this initial stage here, right, this thing, that's where RUBP will bind. This one spot is for carbon dioxide. So how many carbons does RUBP have? How many spots do you see here? No, it's 5. 1, 2, 3, 4, 5. Okay, so remember how I said that there's 5 carbons of RUBP and then 1 carbon from CO2. They bind together to be fixed, to be made into 2 3 carbon chains. So there's a total of 6 carbons that will initiate the reaction. 5 will come from Rubisco. That's right, 5 will come from RUBP, which is a chain of 5 carbons, right? So that's why 5 of these G3Ps are made into 3 RUBP molecules, because how many carbons does 5 G3P have? If each G3P has 3 carbons, 5 of them would have how many? 15. So if each RUBP has 5 carbons, how many can you make using 15 carbons? 3, right? Simple maths. 15 carbons to distribute. Each RUBP molecule requires 5 carbons for itself. So 15 divided by 5, you can see that it will make 3 molecules, which is, yeah, basic maths. So you can actually kind of follow through with the entire thing, the maths of the entire thing, if you remember the initial carbon fixation math. The fact that one RUBP has 5 and carbon dioxide is providing 1, and it makes 2 chains of 2 3-chain carbons. And then the rest of the things happen, right?
Answer: how many 5 carbon RUBP molecules can we make using those carbons? I'm guessing that you figured it out. We can make 3 RUBP molecules. To do this, energy is required. Can you guess where that energy comes from? You said ATP. You are right. Let's watch. Now that we've completed the Calvin cycle for 3 carbon atoms, let's go through the process again so that we can make glucose. Remember, our overall photosynthesis equation has 6 carbon dioxide being used to make 1 glucose molecule with 6 carbons. With this in mind, how many G3Ps are needed to make 1 glucose? You said 2. You are correct. Now you've seen how the Calvin cycle is used to make glucose, and how light-dependent reactions provide the NADPH and ATP that were needed to make the Calvin cycle work. The glucose and other organic compounds made as a result of these processes in photosynthesis are what allow plants and algae to grow and support practically all life, including you. Become an expert on the Calvin cycle by doing it yourself. Play Photosynthesis Interactive, Bioman Biology.
All right, let's move on. All right, anyone still confused about this cycle? Yeah. Okay. So this picture basically is the entire thing, right? It's a light dependent reaction, and then there's the Calvin cycle. I think I've mentioned multiple times. Calvin cycle is another name for the light independent reaction. So you can refer to it as Calvin cycle, or you can refer to it as the light independent cycle. That is up to you. Okay, so let's start from the very top and let's go through until we get to glucose. One more time. So sunlight comes in, that's the first thing required for photosynthesis to occur. Sunlight comes in and it excites the electrons that are found in the photosystem 1, and then these electrons, excited as they are, start to move along the transport chain, going across a few membrane proteins. One of the first membrane proteins it comes across is a hydrogen pump, which will get powered, causing hydrogen to flow inside the thylakoid lumen. So that will essentially cause a higher concentration of hydrogen ions within the thylakoid lumen. So after that, after the electrons are doing that, they will keep moving on until they get to the second photosystem, where they will be used to generate NADPH from NADP+. So the electrons are supplied to the reaction along with hydrogen ions to create NADPH. So the reason NADPH is required is that it carries electrons and hydrogen ions, which can be later used to reduce within the Calvin cycle. So that is the first stage. That's the first thing that happens within the light dependent reaction. The second thing that also occurs is, like I said, the electrons that are being excited need to be replaced, and that's where the H2O is broken down through a process known as photolysis. Photolysis will break down the H2O into two things: hydrogen ions and oxygen atoms, and also take the electrons and supply it to the photosystem. So three things happen essentially: electrons supplied, oxygen is made, and also hydrogen ions are made as well. The oxygen atoms are released into the atmosphere through the stomata, and the hydrogen ions are accumulated within the thylakoid lumen. So far, we know for a fact that within the thylakoid, there's a very high concentration of hydrogen ions. And whenever there's a high concentration, it will always want to equalize with its surroundings. So the high concentration of hydrogen ions will want to naturally flow out of the lumen to go to a low gradient. So the only way they can flow out is through a enzyme referred to as ATP synthase. ATP synthase is also an enzyme. Make sure you guys remember that. So it flows through the ATP synthase, and as it flows through the ATP synthase, it causes the ATP synthase to spin, and that energy is used to convert ADP into ATP, the charged version. So two charged particles have been made, two charged coenzymes have been made, ATP and NADPH. These two are products of this light-dependent reaction. One more product is the oxygen that is being released. So a total of three products are made in this reaction: NADPH, ATP, and also oxygen. So once the three products have been made, oxygen is released to the atmosphere, NADPH is released into the stroma, and it's the same with ATP as well. It's released into the stroma, where the second reaction is facilitated. Remember that the first one requires light to convert all the energy into charged and coenzymes, whereas the second stage does not require light because it already has those charged particles to facilitate the reaction. So it all starts with the second reactant of photosynthesis coming in, which is CO2. When CO2 comes in, it binds to a enzyme known as Rubisco, which is not included here, along with RUBP. When both of those bind together, the enzyme Rubisco will catalyze a reaction where it fixes the carbons to become this first molecule referred to as 3-PGA. So can I get you guys to okay, first let me finish the explanation, then I'll get you to copy it out. So the first molecule that is fixed, that the Rubisco will fix the carbons into, is 3-PGA. The 3 refers to how many carbons it has in its chain, which is 3. that will later be reduced in the next stage, which is reduction, to become into G3P. This reduction is where ATP and NADPH will be used up. The energy from them will be used up, where ATP supplies energy to make a precursor molecule, and then NADPH supplies electrons and hydrogen ions to reduce the precursor molecule into the final molecule known as G3P. This is the key molecule in making glucose. That is the final product. Sorry, that's not the final product, but that is the product of the reactions right before being made into glucose. And this is going to be the same molecule for C3, C4, and CAM. That is what they want to make, G3P. And this G3P, as we know, is a 3-carbon chain. So a total of 6 of these need to be made before the next stage can occur, where 5 of the G3Ps will be sent back into the cycle to be made back into the RUBP molecule from the start through the action of ATP, and 1 will be saved for production of glucose. But since glucose has 6 carbons within it, it requires 2 G3P molecules to even be made into glucose. So this cycle needs to happen twice for enough G3Ps to be made, for it to be made into 1 molecule of glucose. And then we have our final product, which is the glucose. So that is start to finish how photosynthesis occurs within a C3 plant.